asked me today to talk about predicting the future. I'm really talking about what are the disease threats to the Alberta pulse industry. And I guess big question is, how do we know what we don't know? And so how do we predict the future? If we knew exactly what was happening, would we ever run into the disasters that we kind of see occurring worldwide right now, and also that we see in our fields? And we really want to be able to kind of get the balance where we don't come up to this, at this stage here, where we're like, oh, we'll get to that bridge when we cross it, and we get to that bridge and realize that it's pretty much impossible to cross. So we're trying to, kind of predict what's going to happen in the future and come up with management strategies. The problem with pulses is they get a lot of bad diseases. And all the crops do. We see it with canola, new diseases come up. We see it with wheat, new diseases come up. So it's like, how can we balance management in the field with also an eye forward and saying, well, what's going to be a problem in the future? And I think in order to look towards the future, we often need to first look towards the past. And there's this great resource, maybe only for us geeky pathologists that love mining through historical data. But there's this resource, the Canadian Plant Disease Survey, that's available through the Canadian Phytopathological Society website here, that actually has disease records going back 100 years. So this is 1929, but they actually started in the 1920, so we're 100 years in. And then this is what it looks like today, a lot more readable. So what I thought for this presentation was that I would kind of take you through a journey of where did we start with pulses. In, I think, 1978 was maybe the first record that I could find on this Canadian Pulse Disease Survey. How did that inform us as to what was coming up? Did we use that knowledge correctly? And I'm going to give you sort of two different case studies on maybe one time when we should have seen a problem coming, and then maybe now how we can work with that. So what I'm going to start with is looking at field pea surveys. I really struggled for a long time trying to figure out how to make this data exciting, and I really couldn't do it except for just make a table with lots of years on it. But basically, this is just kind of documenting all the field pea surveys that have been done since 1978. That was the first one, when they looked at peas, and that acreage was very, very small in Alberta. And look what the first thing was that they found: root rots. Very, very high incidence. And all they found there was fusarium. And then the other things, ascochyta blight and powdery mildew. So we kind of see that go down through the years. And I would say that the record for pea surveys is not great when you compare it to some other crops like cereals, where we see them pretty much happening every year. So we saw about 9 years of surveys, the same things coming up over and over again. And then there were some surveys that were done just for root rots, kind of into the mid-90s. And every time they were noticing a lot of root rots. A lot of these surveys were done just very regional, so you'll see like northeastern Alberta, you'll be like southern Alberta, central Alberta, Peace River. So there was no like coordinated effort to bring all these surveys together. And then a nice, good news story here with powdery mildew in 2006. That's when we had our resistant cultivars. It was kind of when the last survey was done because we just don't see it in the field anymore. And then when I started in 2011, I looked through this record and was like, okay, well, root rots, we really haven't surveyed for pea diseases in a number of years. Why not? And I think the first proposal that I put forth to APG was looking for root rots. But I was actually looking for fusarium root rots because that's what the historical record told us. Everything was fusarium. And so really this finding of, I'll get to it the next couple slides, but the finding of our new friend here in 2013 really precipitated us doing these surveys every year. So I think we all know the story of our unexpected surprise that we found in 2013. And this was the first report of this destructive pathogen in Saskatchewan in 2012, and then Alberta in 2013. And then what's interesting is that, if you go through the literature, we all of a sudden see this first report of aphanomyces in new areas right around the 2010s into the mid-2010s. So we see it in lentils, first report of lentils in Idaho. And we see the first report of aphanomyces in Saskatchewan in 2012, then our first report in Alberta in 2013, and then first report in North Dakota in, I think this was in 2016. Why all of a sudden this first report of aphanomyces everywhere that peas had been grown? So I think, and I'm going to posit, and this is my hypothesis, that we really had a case of mistaken identity occurring in the province, in really all our pea growing regions for a long time. It was present. If you go through the Canadian Plant Disease Survey, you find it in Manitoba in the mid-1990s. So why didn't we keep looking for it in Saskatchewan and Alberta in that time? But it's really because hindsight is 20/20, as we like to say. And our tools as scientists are only as good as the tools that are available to us at the time. So there's a lot of reasons here why I think we kind of missed Aphanomyces. And so going through this, it's sort of a case study or a SWOT analysis looking at what those strengths, weaknesses, opportunities, threats are, can maybe tell us how we might be able to deal with the next problem that might be coming up on the horizon. And so really there's like a whole bunch of things that just kind of gathered together to create this perfect storm. First is that a lot of the past surveys were based on shoot rating, and they would do some culturing, but it's really, really hard to culture out this pathogen. And then the other problem is with the shoot rating, sometimes the plants actually look quite healthy, and you have to dig up the roots. And I think in my bio I said I'd like to work on foliar pathogens because working with roots is really quite a challenge. You know, we go to survey a field, it takes us an hour to 2 hours to collect that data from digging all the roots and collecting the soil. And so I think they just weren't done as often because of the difficulty in doing them. The surveys were also done in late season because that's when you see the yellowing of the plants. You don't necessarily see it early on, but as we've been doing more and more digging and looking at the progression of pathogens, we really see that Aphanomyces hits its peak in mid-June. And then the surveys were done usually early August. You've already missed it there. When you do culture it, Fusarium just takes over the cultures. And so that's why when we look at the historical record, all we see is Fusarium coming out from culturing. It takes a lot of time to actually pull it out of the roots and isolate it, usually about 3 to 4 months to try to make sure that you've dotted all your I's and crossed all your T's and make sure you're doing it right. And then at the time, really why we were able to find it so rapidly, 2013 onwards, was because of DNA tests. And at that time, we just didn't have that DNA testing technology. And in fact, a new test was only developed in 2014. So it was really kind of serendipitous that at the time that we were looking for Aphanomyces, a group in France had just published this new test that allowed us to find it. We are kind of bound by the technology available to us. And when we started looking for Aphanomyces, one of the first things that we did was say, well, there's all these advances in genomics. Why are we spending our time isolating, culturing, using one DNA test at a time? Let's use genomics and look at the whole pathogen complex that's in that soil. So we collected soil from Alberta, Saskatchewan, and Manitoba that we knew had really bad root rot. We used the genomics tools that were available to us. So this is DNA sequencing tried to DNA sequence the whole pathogen complex within the soil. And we use the standard barcode that all scientists use to be able to figure out what species you have. And if you look down here, there's no Aphanomyces that comes up. It's all mostly Pythium, and we all know Pythium is a pretty standard pathogen in most soils. And so this is with the new tools that are available to us, and we're still missing it. So what we actually had to do was we had to know already that Aphanomyces was in the soils in order to develop a tool so we could detect Aphanomyces in the soils. So you kind of already have to know what you're starting with. And so we changed the little barcode here in the gray bars. So we just tweaked this barcode a little bit so that it favors Aphanomyces a little more than it favors Pythium. And then we can see that Aphanomyces is all of a sudden popping up in our soil samples, and we find it at about 20 to 30% abundance of this pathogen complex in the soils. So the tools, so it's you have to be biased in order to do science, but scientists were supposed to be unbiased, so it's hard to look at the whole picture. And then what happened here is that I think everyone has probably seen this graph before, this map, is that for a new pathogen detection in 2013, we really saw that Aphanomyces was present everywhere peas and lentils had been grown. So positive pins are the reds, positive yellows are lentils. So positive red pins are peas, positive yellow pins are lentils. So the hypothesis now, and I said, going out on a limb to say that we probably had this pathogen for a while, we just didn't have the tools to look for it, and we maybe didn't use our crystal ball well enough to predict what was happening in the future. So I'm going to contrast this with a new up-and-coming story in dry beans. And so when we look at the dry bean surveys, again, the first one was done in 1978 to 1979, white mold and bacterial blights. And that's basically all we see for the next, however many years that is, just about 40 years, right? White mold, bacterial blights. That hasn't changed. What's changed is that if you look kind of far back into the past, white mold was pretty much severe every year that you looked at it. And then when we started looking for it again in 2015, 2016, 2018, it's actually been pretty low to moderate compared to what we saw earlier. So this might be, we're going to dig into this a little bit more and maybe try to write a review paper. But I think it's a bit of a good news story with some of the cultivars that have come out of the Lethbridge breeding program that have some better standability against white mold. And then you can see other things, funny things pop up, like bacterial wilt and pink seed, and then they just kind of go away. So, but the question is, have they really gone away? And I'll show you that in the next slide. But then what's interesting is that we started looking for root rot because we knew it was a problem in peas. We wanted to know, is it a problem in bean? And it was actually quite low. But we'll get to it in 2 slides why this is maybe not correct. So just the story with bacterial wilts and why we have to pay attention to what happened before. They kind of went away. I think there was a big peak. Everyone wanted to study them, and then it just went away. And you're like, oh, well, why did it just go away? And we still don't actually know why it kind of went away, and then now it's coming back. And really what it's coming back is in our yellow market class, which are a very high-value yellow bean, and they just seem to be more susceptible to it. And so we just looked at seed contaminated from Idaho or Alberta with a whole bunch of different pathogens, bacterial pathogens, and really what we find is that we find that most of them in these yellow sources that are grown in Alberta. So what that tells us is that we have to maintain our good management practices of using seed grown in Idaho to reduce the risk of these bacterial diseases popping back up and to remain vigilant on what's out there. Now the other thing that I want to talk about is this new threat, and I think those that were at the Viterra dry bean meeting in February may have already heard this. This is the very short Coles note version of what we're kind of looking for. So there's a new threat, Phanomyces in beans. So we went looking for it for 3 years, 2015, 2016, 2017. Didn't find it at all. It's like root rots are not going to be a problem for dry beans in southern Alberta. And then in 2019, Viterra sent me all these samples of bean roots that had root rot and said, well, what's going on? I'm like, well, it can't be a Phanomyces. We don't have a Phanomyces in beans. Fortunately, we have that nice DNA diagnostic tool now. So the first thing we did was just tested these roots to see if they were positive for Phanomyces, just because it's what we screen for in peas. And very surprisingly, it was positive. So we went through the whole step of isolating, getting it into isolation. It's, like I said, this takes about 2 or 3 months. We're actually just finishing this process now, and we got these samples in August. So it takes a long time to get through this process of isolating it out. I don't know why that didn't show up. But what's interesting here is that this isolate that we pulled out of dry bean, we inoculated it onto peas and dry bean, and the peas look perfectly healthy. And actually what we had done before when we were looking for dry bean was that we had tested all of our soils against peas, thinking peas were the more susceptible host. But the dry bean isolate seems to be very specific to dry bean and doesn't infect peas. And then we just, we confirmed that by doing some DNA testing of the culture and the roots. Now I should say, us pathologists are very, very cautious folks. We don't like to say something until we're 100% certain. So I'm 95% certain right now that it's a Phanomyces. We have to send this off for sequencing, and then we get this whole DNA sequence back, and then we have to compare it to a known sequence to make sure that we're not misidentifying something. So we are just waiting on this last step, but all the other signs are pointing towards a Phanomyces in beans. But I think we're in a different position than we were in peas. And like I said, in 2015, 2016, 2017, we went looking for root rots, and it was really, really low. Like our severity of 1 to 7, we're below a 2. So that means there's like a tiny 2-millimeter lesion on the roots for the most part. And then we tried to use our DNA diagnostics to look if there was a Phanomyces. Very, very low levels. I think this actually is about 1. It comes out to 1 in 17 fields or something like that. And we diagnosed those with DNA, but we were never able to do that whole 6-month confirmation thing. So, I thought, oh, well, the industry's safe. But I think part of the problem is these surveys is that it is a little bit like looking for a needle in a haystack. We wanted to, we were trying to be proactive and say, is it there? But we were surveying 20 to 30 fields when you have 45,000 hectares of beans, right? So it's really these close ties with industries, agronomists, et cetera, that alert us to new problems that I think are the key to our crystal ball. So when we find a new threat, then how do we deal with it? And this is where I think it's a big contrast from what happened with peas, where we found it, it was everywhere. With beans, what we're hoping is that we found it in a couple fields. We can be a lot more vigilant now about looking for it and making sure that we then introduce recommendations and management practices to reduce that spread outwards. And so what I'm hoping to do is monitor dry bean fields, and then also we don't know. There could be a lot of differences in cultivar screening. We just don't know that yet. But we're at a much better position here to deal with an upcoming threat than maybe we were with peas, where it kind of took us by surprise. I'm just going to talk very briefly about lentil and chickpea, basically just to say these crops may be being underrepresented. It's been a while since there's been surveys. We went in lentils, we've been looking a lot at root rots, but we haven't really looked at anything else. And then the problem is I got my hands full with peas and dry beans. So, just sort of capacity in the province to be able to deal with some of these pathogens. Seems like there was a lot of fava bean growers in the room. And so interestingly, the only time fava bean surveys were done were 1978 to 1979. And I guess they weren't grown for a long time, and now they're coming back up. And alternaria leaf spot, powdery mildew, I was actually surprised to see that because I hadn't heard of any powdery mildew spots. Ascochyta blight, chocolate spot, and root rot. So from 2017 to 2019, we have actually been going out and surveying. We were looking, the primary purpose for the project was to look for chocolate spot, but doing our due diligence, we're like, well, we're going to isolate everything that comes off those leaves and actually see what our different pathogen risks are. And so chocolate spot is caused by botrytis right here. We can see that it's actually fairly low percentage that's coming off. About 8 was our lowest, up to 15. And then we get these other things like alternaria, fusarium somehow is on the leaves, sclerotinia very, very low, and then stemphillium pretty high. So we followed that up with doing some pathogenicity tests. And I guess this is the question now, are these going to be our new threats? Alternaria, we find, I think it was like 35 to 40%. It's not super pathogenic, but it's definitely causing some problems on the fava beans. Stemphillium is one that comes out a lot, and it's actually fairly pathogenic as well. And then what's interesting here is Colototricum, very, very low percent. I think it was only like 0.1 to 1% that we found it, but it's very, very pathogenic, very virulent on fava beans. So it's something that is maybe not a problem now, but we just have to look forward into that future and think, okay, will this become a problem as fava bean production increases? With the last two slides, I really like what Jen was talking about this morning of this integration between scientists and all different aspects of the sector. Because AAFC has this new working white paper on what we're calling biovigilance. And sometimes you just kind of get policy things that you kind of scratch your head about. Oh, wait, I'm on camera. But other times, this is something that was put together by scientists as a new framework to really assess risks and kind of biosecurity issues to Canadian agriculture, but also how we deal with our current issues. And I like it because it brings in every part of the sort of value chain thing, starting with awareness, which is really what our surveys are about, but it's taking those surveys and integrating them into the rest of this biovigilance continuum to make sure that our surveys aren't just going into a black box somewhere that a plant pathologist 100 years from now looks at and says, oh, that's what their problems were that they had then. They should have known better. So I think it's really moving those surveys forward through the rest of this and making sure that we are addressing those threats to the value chain. So in the end, we're like, can we predict the future? Not really. I think it's only as good as the tools currently available to us. And the tools are continuously changing and making so we have better and better decisions, but you still have to know where to look. And as I said, surveillance is that primary tool, but we need to integrate it into every other part of the spectrum. Pay attention to the bigger picture, what's happened elsewhere. I do think we had our head in the sand a little bit about Aphanomyces and should have looked into that history and said, everywhere they've grown peas, they've had a problem with Aphanomyces, and we could have maybe started looking for it a little bit earlier. The other thing is, like I said, when I look for things, I'm looking for a needle in a haystack. I'm not out there in all the fields. I am completely open to people calling me, emailing me, tweeting me if they see something that's unusual in their fields. If you call me, where's Peter? I might not call you back. But if you email me or tweet me, I usually do get back to you, but I'm not very good at returning calls. But, and I think, I try to keep an open communication with the farmers, agronomists. It's been really good with the Viterra dry bean industry because they come to me with a lot of problems. And I think that's really how we can predict the future and find those needles in the haystack, is to make sure that we have that open communication.